Hello everyone! I appear to be stuck waiting in my car for my wife for a brief period of time, so I figure, why not, at the very least, start recording this lecture. So, it may suddenly not be in the car, but, whatever. Let's see what happens. So here we go. This is lecture number 15 on the chromosomal basis of inheritance. The previous lecture was all about good old Mendel and his rules of inheritance. We even talked about some non-Mendelian inheritance, things like codominance, pleiotropy. And now we're going to get into how it all works when you add in the fact that our genes are on chromosomes, right? An allele, as we learned in Mendelian genetics, is the genetic unit, but genes are in DNA. We talked in class about how we know that it's DNA. Good old Oswald and Avery and Hershey and Chase and their experiments that showed genes are in the DNA. And the locus of every gene is on a chromosome. So when we talk about inheritance, when we talk about genetics, it's not individual genes sort of shuffling around on their own, but rather the genes and their entire chromosomes that are inherited. So when we talk about inheritance, it's important to talk about chromosomal inheritance and how that can affect the general rules, right? So first, we have the law of segregation. Remember, in the law of segregation, the alleles segregate independently of one another so that we can have multiple different combinations of genes. There are, in fact, many genes on a single chromosome, right? A single chromosome has, if it's a Y chromosome, I'm not entirely sure, but I think it's less than 100, to thousands of genes on normal chromosomes. And so it's the chromosomes and all of their genes that actually segregate. During meiosis, when you remember, the chromosome from mom and the chromosome from dad could end up on either side, and then they're homologous and they're pulled apart. Or, right? Independent assortment. And the law of segregation says they segregate independently, like the genes, the alleles segregate independently of one another. So now it's the chromosomes and all their genes segregating independently from one another. It still results in Mendelian genetics following those basic rules. But when there's neat things going on with chromosomes, it's important to add those in. So let's talk about humans. First off, let's talk about red-green color blindness and why it's in this chapter. It is, in fact, Mendelian, right? We have Mendelian inheritance with red-green color blindness. It's recessive, so red-green color blindness is the recessive allele, and color vision is the dominant allele. So say you have a colorblind mom and a dad who can see in color, right? And they decide to have kids. Well, let's go through some basic Mendelian rules of inheritance, and let's play a game of what genotype is dad, right? So let's do some Punnett Squares with the rules of Mendelian inheritance. Here we go. Red-green color blindness. Dad, let's see what happens if he's homozygous dominant, and let's see what happens if he's heterozygous. What ratios would we expect to see in these two situations? So first off, it's recessive. So mom having color blindness means she's homozygous recessive. And there we go. We put her 1, 2 homozygous recessive alleles there. Nice and independent because a gamete only gets 1. And then with dad right over there, separate them out. This is our example of what to expect if he's homozygous dominant. And boom, there we go. Oh, a heterozygote. A heterozygote. You know what? All heterozygotes. So, if dad is homozygous dominant, then the phenotypes, the phenotypic ratio is basically 1.0 of all color, 1 to 0. They all see color vision. And the genotypes are 4-0 heterozygous. All right, how about our other one? What ratios for when it's a heterozygous dad times a colorblind mom? Here we go. Boom, heterozygous. Oh, homozygous recessive. There's someone colorblind. Okay, so we see a 50-50 ratio, right? We would have 2 offspring that have color vision and 2 colorblind offspring with that same 2-to-2 ratio, or 1-to-1 if you like to reduce, of the genotypes as well. 2 heterozygous to 2 homozygous recessive. Okay, so knowing these, what if I told you that a cross would result in a 50-50 ratio? What would be Dad's genotype? Huh? Anybody? That's right. He would be expected to be heterozygous. Well, here's where it gets neat, because anytime you have a father with color vision and he has a child with a colorblind mother, you always, 100% of the time, see a 50-50 ratio. That's kind of weird. That's kind of weird. No matter how many times, how many offspring, no matter how many times you cross the parents, how many offspring you produce, you'll see overall a 50-50 ratio. Here's where it gets even more fun. All daughters have color vision. They can see red and green. And all sons are colorblind. So what in the good fuck is happening here, right? Huh? That's weird. Well, remember where we are. The chapter on chromosomal inheritance. What chromosome does Dad give his sons? What chromosome does Dad give his daughters? Because we know Dad is carrying the allele for full color vision, right? So, what chromosome is involved here? The sons are all colorblind. Some of you may have gotten it right. Some of you may have said the Y chromosome is carrying the vision gene. But we run into a little issue here. Mom's colorblind. How many Y chromosomes does she have? Yeah. So it is, in fact, carried on the X chromosome. Colorblindness, the allele for colorblindness, the gene for colorblindness, is on the X chromosome. It's what we call a sex-linked gene. Specifically, it's an X-linked gene. So sex-linked genes are a gene that is carried on either the X or the Y chromosome. And it results in some funky stuff when dealing with, humans, because we're either XX or XY. Colorblindness itself is X-linked. So now that we know that, let's put our colorblind mom and our color vision dad together on a Punnett square and see how this works. All right, so here's a daughter, and she's heterozygous. And there's a son, and now we can see why the son is colorblind. In order to be male, he has to get dad's Y chromosome. The Y does not have a gene for color vision on it. So the only color vision gene he gets is mom's recessive colorblind gene. So even though dad has color vision, the sons are colorblind. There we go. So you get that 50-50 ratio where the daughters have color vision and the sons are colorblind. This happens on X-linked genes. It's really cool. So 2 are female with color vision, 2 are male that are colorblind, so the phenotypic ratio is 2-to-2, and the genotypic ratio is 2-to-2, with 2 being XX color vision heterozygotes, carriers for colorblindness. If you'll recall, a carrier is someone who has the allele for a recessive disorder, but does not display the recessive disorder. So the daughters have 2 X chromosomes and are carriers, whereas the sons fully express it. Boom. Let's talk about some other X-linked recessive disorders. Hemophilia is an X-linked recessive disorder. Kind of cool. In hemophilia, you're missing clotting factors. What this means is that your blood clots slowly, or if you have it quite bad, it clots basically not at all. We already did colorblindness. We know that's X-linked. Muscular dystrophy, you might have heard of it, is X-linked. So these are all recessive disorders inherited on the X chromosome. So, hemophilia is cool. It was once called the royal disease. It's X-linked, and Queen Victoria had children that expressed hemophilia, right? So, her husband did not have hemophilia, and Queen Victoria herself was a carrier of hemophilia. So she actually did not show the disease very much. There can actually be variation in how much you express hemophilia due to the fact that you have a little bit of codominance, I think, happening, and women have another thing going on with their X chromosomes, which we'll get into. So anyway, all her sons had hemophilia, which sucked quite a bit. But even her daughters were carriers of statistically speaking, up to 50% of her daughters were carriers of hemophilia. Now, this ended up a disease that affected multiple royal families all over Europe. Why, you ask? Well, anybody watch some Game of Thrones? How does royal diplomacy work? Robb Stark was gonna marry someone. That's right. So, intermarriage was how you did a lot of diplomatic relationships way back in the Middle Ages through the 1700s and 1800s, I think. Oh, that stopped. Anyway, so Queen Victoria's carriers of hemophilia, and her hemophiliac son, I think her hemophiliac son might have died, but anyway, her daughters carried it and married into other royal families. And then those royal families picked up carriers of hemophilia, and some of their children expressed it. So hemophilia actually became a pretty rough disease in medieval Europe, the royal disease. The cool thing about hemophilia is that it's fully treatable now. We've isolated the clotting factors that are missing, and we can pick up clotting factors when people donate blood, and then just transfuse them into people who have hemophilia. So if you have hemophilia, nowadays you just receive regular transfusions of clotting factors, and your body works pretty much normally. Okay. How about Y-linked disorders, right? Sex-linked isn't just all the big old X chromosome. How about the weird and fanciful Y chromosome? Hypertrichosis of the pinna. Hypertrichosis of the pinna. Well, let's go with Latin. Anybody know pinna? Hyper, lots. How about trick? This is one you might not see all that often. Hair. It's of or relating to the hair. Hypertrichosis of the pinna, excessively hairy ears. So if you have hypertrichosis of the pinna, all of your sons will have hypertrichosis of the pinna. All of their sons will have hypertrichosis of the pinna. You will pass hypertrichosis of the pinna down as long as your family line exists. Only the daughters are safe. So, there's that. The other thing you can see that are Y-linked are sex differentiation disorders, although these tend to not be something we look at as inherited. The TDF gene is the gene on the Y chromosome that triggers early testosterone production, and that triggers the embryonic development pathway that leads to a male fetus. So if you have mutations in the TDF gene, you can result in disorders of sexual differentiation. And that tends to really cause infertility. No matter how it manifests, infertility tends to be the result. So this is not really something, as much as I hate saying this out loud, that people have to worry about passing on to their children, because they're not having any. So why are there so few Y-linked disorders? The Y chromosome is tiny. It's almost useless. It produces a male, and that's about it. He's so small. He's so very small. Not a lot of genes on there. Apparently, it's just genes for your hairy ears and having a penis. All right. Next up, a cool thing that affects how genes are expressed in women. All women have Barr bodies. What's a X chromosome like you doing in a bar like this? Which is much funnier when you understand what a Barr body is. So first off, we know that an X chromosome is required to live. A fetus is non-viable. Well, an embryo is non-viable if it does not have at least one X chromosome. There's information on that needed to be alive. That's why you are XY or XX, no YY. So, how many X chromosomes are required to be alive? So, in other words, how many X chromosomes does a woman have? 2, right? Does she need both to be alive to see this, to answer this? Let's look at good old men. How many X chromosomes do they have? Just the one, XY. A cell only needs one functioning X chromosome in the nucleus in order to perform all its functions. So, what happens to the spare X chromosome? Well, I think you're getting it at this point that it has to do with the phrase Barr body. A Barr body is the result of random inactivation of an X chromosome at some point during embryonic and fetal development, right? So, the zygote, when a sperm fertilizes an egg, you get the first cell of the new generation, the zygote. It has 2 X chromosomes, and it's going to start undergoing mitosis. And you're going to develop a large number of cells. And eventually those cells are going to start to differentiate. Now, initially, those X chromosomes are not being randomly inactivated. But at some point during embryonic development, especially during the cell differentiation period, when the cell lines that will produce the nervous system differentiate from the cell lines that will produce the muscles, and those differentiate from the cell lines that will produce the gastric system, right? So you have differentiation, cell lines essentially becoming more specialized. And they differentiate multiple times, becoming more and more specialized throughout development. And at some point during embryonic development, X chromosomes are inactivated. You get to the point where you only need the one functioning X chromosome. The other X chromosome, so it happens during embryonic development, the inactivated X chromosome actually sort of compresses into a small little hunk of DNA almost. So the linear strand of DNA that corresponds to the X chromosome coils up and compresses so that none of its DNA is easily available when trying to read the DNA and produce proteins off of it, right? Now, it's a 50-50 shot. It is random. You have 2 X chromosomes, one from mom, one from dad, and which one gets inactivated is completely random. So this can result in some interesting stuff. For instance, women tend to show varying degrees of hemophilia, in part because it's codominant, and if you have one functioning allele and one hemophilia allele, your hemophilia isn't going to be as bad, simply because you've got a dominant one compensating. And even if you're homozygous recessive for hemophilia, one of those alleles, no, I'm lost my track here. This is what I get trying to do stuff at night. it is really when you're heterozygous. Damn it. So even heterozygotes who have some compensation due to their dominant allele will show varying degrees of hemophilia, in part because it's codominant and in part because in like roughly 50% of the cells, the dominant allele got inactivated and the recessive allele remained active. So the dominant allele became the Barr body. The Barr body is the compressed inactive X chromosome. It goes from, a nice linear piece of DNA here, and, we go, oh, let's read this gene and go make a protein from this gene. Ooh, we're going to do that. And then it curls up, and, oh, no, we can't read that gene. But over here is the other X chromosome. We'll read that gene. So if this is the dominant gene, it may get compressed, and all you have is the recessive one, or vice versa. So you'll see varying degrees of hemophilia based on what bone marrow X chromosomes have the Barr body. Hopefully that didn't get too crazy in my brain, but I can explain it again, this time using cats, because everybody loves cats in the internet age. Cats are pretty convenient because black fur and brown fur are carried on the X chromosome. Both the genes for black fur and the genes for brown fur are carried on the X chromosome. So here we have the zygote, and this kitty is heterozygous. On one chromosome, it has the gene for brown fur, and on the other chromosome, it has the gene for black fur. And at some point during embryonic development, we start to see Barr bodies in activation. So we can see in some of them, the brown fur allele gene is inactivated. The brown fur X chromosome is inactivated. And in others, the black fur X chromosome is inactivated. I have it, a nice solid dot. So eventually, those different differentiated cell lines will go on to produce skin. Now, multiple skin stem cells produce the skin. And so you can have a large number of different skin stem cells that have different Barr bodies. They aren't all derived from a single embryonic cell that had a single X chromosome inactivated. Multiples of these went on to differentiate into the skin cell line. I ran out of space, so had to do this at like a 8-cell embryo, which is not when inactivation occurs. It's just, a nice drawing I made. So, anyway, wait, did I make this? I might have stole this from the internet. Who knows at this point? Anyway, different skin cell lines are derived from different embryonic cells that had a different Barr body, and what you get are torties. What color is a torti? A mix of black and brown fur. Appears orange to a lot of people. I would call it orange, but that's considered the brown fur. So there's a torti, and you can see that the pattern is almost random. You would be very hard-pressed to find two torties with the exact same pattern. And that's because it was random which cells had an X that inactivated. It was random which X inactivated in the embryonic stem cells. And the skin cells that were derived also had that random inactivated X. So the black fur lines are derived from embryonic cells that inactivated the brown, and the brown fur is derived from stem cell lines that had the inactivated black. If you want to know what a calico is a tortie that has white. So if you have a tortoiseshell cat that has white fur, in fact, that's a calico. The same rules applied to its black and brown patterns. Now, here's something fascinating. You might actually know this already if you're into cats. What sex is a tortie? Always female. You don't see male torties, and that is literally because it requires two X chromosomes with the formation of Barr bodies in order to get that pattern. If you have a male tortoiseshell cat, that male is carrying two X chromosomes. And that would be extremely rare, but it has happened. So, Barr body inactivation and torties. So, there you go. Beautiful. It's brown and orange in random patterns because at some point during embryonic development, different lines that produce different parts of the skin had either the black fur or the brown fur X chromosome inactivated, turned into a Barr body. Super cool. And I got ahead of myself because I already explained why they're almost always girls. Okay, so fur color, X-linked. If you have torties are heterozygous for black and brown fur. Black X, orange X, and so on. Well, I already explained all that. So, I guess I could repeat myself, but good Lord, I already did that. Right? So remember, tortoiseshell cats have fur color that's X-linked. They carry a gene for black fur or a gene for brown fur. And a heterozygous cat that has a black fur and a brown fur gene will produce a tortie. So, blurp. Okay, now let's talk about the next thing, linked genes. Okay, so linked genes are genes that occur on the same chromosome. However, that's not what makes them linked. What makes them linked is their extreme closeness to one another, right? This means that they're almost always inherited together. If you say, well, they're on the same chromosome, so kind of they're always inherited together. Remember, crossing over happens a lot. In women, on average, in one meiosis event to produce eggs, it occurs 72 times in 23 homologous pairs. Crossing over happens a lot. So when we say genes are linked and they tend to be inherited together, what we really mean is that while they're not physically like attached or hydrogen bonded to each other in some way, they are close enough to each other on the chromosome that crossing over extremely rarely separates them. Right? They're so close together that it would be statistically very unlikely for crossing over. Like if you have, genes right here and then crossing over, it might occur here or 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 here, but very unlikely that it'll hit right there to separate them. Those genes would be linked. Whereas if you have genes on the chromosome here, crossing over could occur here or there and separate them. So the closer they are together, the more likely they are to be inherited together. When they're close enough that crossing over only extremely rarely separates them, they are considered linked genes. And that's all we have on linked genes. So now let's talk about chromosomal disorders. Okay, so we talk about chromosomal disorders. We're going to be mainly talking about two basic things. What happens when we lose and reattach DNA to chromosomes, and something we call aneuploidy. We'll start with aneuploidy. Aneuploidy means we have a different number of chromosomes than the diploid number. So we have an abnormal number of chromosomes. Monosomy is where you are missing a chromosome. You're down one chromosome, 2N minus 1. In reality, what we're talking about here, remember, every chromosome comes in pairs, one from mom, one from dad. So in one of your chromosomes, either the egg or the sperm didn't carry it. So either the egg or the sperm didn't have the chromosome. So if you have, the sperm and the egg, one's missing, then you only get one, and you're 2N, which is the diploid number, minus 1. You only have one of a given pair. You're missing one. Turner syndrome is a woman who has only a single X chromosome. Turner syndrome. You generally, with Turner syndrome, a woman is shorter, much shorter. She has very underdeveloped, if at all, breasts. She tends to have a much higher pitch voice, generally infertile. So, Turner syndrome, without that second X chromosome, for some reason, a woman doesn't develop through puberty very well. There are some other physiological symptoms associated with it, but a lot of it is sort of a failure to go through puberty physiologically. Speaking of sex differentiation wise. So anyway, yeah, Turner syndrome. Then we have trisomy, 2n+1. If monosomy means we're down one chromosome, where in one of your 23 pairs you only have one instead of two, then trisomy means in one of your 23 pairs you have 3 instead of 2. So you ended up with an extra chromosome. Trisomy 21 is called Down syndrome. You're all familiar with Down syndrome. XXY is Klinefelter's syndrome. So another example of a trisomy. So fascinating. There was once in the 70s, I think, either the 60s or the 70s, for a little while, there was this idea that the Y chromosome is where you triggered male development and testosterone production. So men who had 2 Y chromosomes must be like larger and more powerful and more aggressive. And they thought maybe XYY men would end up being like super manly aggressive people. And they even hypothesized that they'd be like overrepresented in the prison community, because if they're super aggressive and random, they're more likely to get in fights and assaults and end up in prison. Supermen, the super Y chromosome people. As it turns out, that was all bullshit. No. XYY did not produce like super masculine ultra men who got into fights and beat everyone up and were criminals. It just turns out to just be XYY. There seemed to be no real sociological impacts of having a second Y chromosome. Okay, let's talk about how we end up with aneuploidy. So the process of either losing or getting an extra chromosome occurs through non-disjunction, right? So if you have a monosomy or a trisomy, it means that an egg or a sperm either had one less, in the case of monosomy, or one more, in the case of trisomy, chromosome than normal. So the process that leads to a gamete having one more or less chromosome than normal is called non-disjunction. And non-disjunction is simply a failure of chromosomes to separate. A failure of chromosomes to separate. It occurs during meiosis, during the production of the chromosome. It can either happen in meiosis I, so anaphase 1, or meiosis II, anaphase 2. Because remember, in both of those, that's where you're doing the separating. In meiosis I, in anaphase 1, what separates from each other? Huh? What was that? Did you say homologous pairs? You're right, genius. The homologous pairs separate from each other in anaphase 1. So non-disjunction during meiosis I means the homologous pair failed to separate. So one of the 23 homologous pairs in humans failed to separate. In meiosis II, what separates? What is it that's cleaved in half? That's right. The centromere is cleaved in half, and the sister chromatids separate. right? So if you have non-disjunction in meiosis II, then the sister chromatids have failed to separate. And we'll pick this up when I get home. Control-Shift. Honestly, at this point, I have no idea. I think it was about meiosis II. I was saying that non-disjunction in meiosis II, the sister chromatids fail to separate. So the centromere doesn't break. And when the centromere doesn't break, the whole shebang just ends up going to one pole. So that's what happens in meiosis II, in anaphase 2 of meiosis II. So you get aneuploid gametes, a gamete that is either N-1 because they're haploid or N+1. So meiosis I, you fail to separate the homologous pair. So instead of our homologous pairs separating and moving to one end, boom, the whole homologous pair moves there. And you get, as an ultimate result of this, all of the gametes produced are aneuploid, right? Because if you think about it, once we have two, we have the pair, the homologous pair, and they move to one end, now you have one end that's got two chromosomes and one end that's got no chromosomes. So when that separates into two cells at the end of meiosis I, you have one that's N-1, one that's N+1, and then when they go on, you'll get, at the end of meiosis II, two of them that are N-1 and two of them that are N+1. And I have a picture to show it. Then meiosis II, here your sister chromatids fail to separate. So the centromere doesn't break. Normally the centromere breaks and they move to opposite poles. Instead, the centromere fails to break. And so because it's meiosis II, this is occurring in one of the two cells produced in meiosis I. Right? So meiosis I, you have the one big cell and it goes through and you get homologous pairs and they separate, and then you have two haploid cells that have intact, sister chromatids in there, but they're still haploid here. And then they go on to meiosis II, and each one of those goes into 4 cells. Well, generally non-disjunction is rare enough that it's only going to occur in one of these two. And so when non-disjunction occurs during meiosis II, the sister chromatids in this one of those sister chromatids fails to break, and instead the whole thing moves to a pole, and that one cell where it occurred, you'll get N+1, N-1. Whereas this other cell where it didn't occur, you'll get 2 regular haploid gametes, N and N. So 2 N cells, 1 N+1, and 1 N-1. And here's the picture to show you that. So here's non-disjunction in meiosis I. In anaphase I, the homologous pairs fail to separate, and boom and boom. So this right here, this pair is the homologous pairs failing to separate, and here they're absent. And so because they're absent, we're N-1, and because we have, that extra set, homologous, chromosome 1 of homologous pair, and then the second chromosome of the homologous pair, we get N+1 in 2 cells. Whereas here, it only occurs in meiosis II, non-disjunction and anaphase, and the centromere doesn't break, so the sister chromatids fail to separate. And in the cell in which it occurred, you get N+1 and N-1, and in the other cell, you get regular haploid gametes, N. Okay, last thing is talking about recombinant chromosomes. So in recombinant chromosomes, we've either got new DNA attached to an intact chromosome, a chromosome that has lost a section of DNA, or a chromosome that lost its DNA, a section of DNA, and then had that section reattach, but in a different way. So it is not identical to the original. Oftentimes this does not occur on the original chromosome. Like, it doesn't just disconnect and then reattach on the original chromosome in the same spot. It'll either get messed up, reattached and messed up, change direction, something like that, or it'll end up on a new chromosome. The original chromosome will have just permanently lost some DNA. So it does not end up in the original orientation. A recombinant chromosome does not have the normal genetic makeup. So I actually have pictures to help me explain this. So the first thing we see, in order to have a recombinant chromosome, a chromosome has to experience a deletion. So a deletion is where a portion of the DNA is excised from the chromosome, or a number of genes. So you have your chromosome, and just a section of it gets cut out. It's just cut out and lost. And so a recombinant that's had a deletion is now missing genes. So what happens to the missing DNA is what produces the other types of recombinants. So what happens when that broken, that excised bit goes somewhere else? Okay, so it could bind to a sister chromatid, and we could have duplication. So remember, a chromosome is, an X-shaped, right? So say you lose a portion of the genes, like this section, maybe they attach right here. And now you have, since they're sister chromatids and identical, this section is identical to this section, it attaches, and now we've got duplicate sections. Finger puppets are terrible ways to explain this, but the pictures are coming up. Sometimes it could reattach to a new chromosome backwards, or it could reattach to its original backwards. And when it reattaches backwards, we have an inversion. And that excised portion from the deletion could end up attaching to a non-homologous chromosome, at which point it's packed up its own little sandwich and it heads down the road and it goes, screw you, I'm finding someone who cares about me, and binds to a completely different chromosome. So it will translocate somewhere else. So here we go. This is a deletion. Here's our chromosome, A, B, C, D, E, F, G, H, I. And D, E, F is excised, cut out through some process. And boom, we've lost that. So here's our chromosome that's had a deletion occur. And this is now a recombinant. It's got a deletion. Here's an inversion, right? The part gets excised and reattached backwards. It's inverted. So it goes from DEF to FED. Here we have a duplication. So these are identical sister chromatids, A, B, C, D, E, F, G, H, I, right? And the DEF gets cut out here and then manages to insert and bind to its sister chromatid. So it gets cut out and we get a deletion and it binds over here. So we end up with a portion deleted and we get a replication. Let's see, how did I do this? Apparently I went with the whole bottom portion getting cut out instead of DEF. So forget what I said about DEF. Here we have two identical sister chromatids and the bottom portion gets cut off. Excised, there's a deletion of FGHI from this second chromosome. And here's that poor second chromosome. And instead, FGHI ends up reattaching completely. Now we have ABCDEFGHI, and then a duplication of FGHI. So it was lost chromosome, the first sister chromatid in this pair, had it deleted, and it bound to the next sister chromatid. And so that resulted in a duplication of genes. We've doubled up the particular genes. So You'll notice all of these have a deletion involved. So, translocation. We have A, B, C, D, E, F. And over here is chromosome WXYZ. And DEF is lost. Deletion from this chromosome. And then it translocates to the WXYZ chromosome. We have WXYZ DEF. So, all of these are recombinants. We have the deletion recombinant, where we've lost a set of genes. We have an inversion recombinant, where a set of genes was excised and then reattached, but in reverse order. And then we have a duplication recombinant, where one of the sister chromatid pairs had genes excised, and it attached, those lost genes attached to the identical sister chromatid, resulting in duplicate genes, hence duplication. Then translocation, we have one deletion recombinant and then a translocation recombinant right here, where it got extra genes. And that's it. So that's actually the end of the lecture as well. So I hope you guys found it informative, and y'all can have a good day, night, whatever. Peace out. Biology